For attendees, thanks for joining us for the round-the-clock journey to highlight the project to map the human body at multiple scales. For those who might just be joining, the Human Reference Atlas project aims to map all the cells of the human body to advance biomedical research and clinical practice. But mapping the cells is part of a bigger story and how cells interact and ultimately create us. So for this tour, we'll be exploring ways we might bring the Human Reference Atlas across scales to the public. We'll explore the kinds of ideas that might inform an exhibit at a science museum or other cultural institution, some of the challenges and opportunities in bringing the human body at multiple scales to the public, how it might be experienced across sensory and intellectual domains. There are a variety of ways informal science learning through hands-on and embodied exhibits, visualization, modeling, and other innovative modalities can both inform the public of the value of the work and provide new perspectives and tools to advance biomedical, educational, and artistic professional fields of endeavor. So we've invited 4 experts from diverse fields to participate with all of you over the next hour to discuss their work and help us imagine how to get these ideas into the hands and minds of the next generation of explorers at one of the most intimate and challenging frontiers of science ourselves. I'll introduce each panelist and they'll provide you with a brief overview of their work. Then we'll explore as a group some of the big ideas and questions relating to the public multi-scale human body experience, and then address some of your questions, which may be put into the Q&A section of the Zoom interface. So I'm Steve Uzzo from the National Museum of Mathematics in New York City, and I'll be moderating the panel. So our first panelist is Weidi Zhang. I hope I'm pronouncing your name correctly. A new media artist and designer and assistant professor of immersive experience design at Arizona State University. Her interdisciplinary art and design research investigates a speculative assemblage at the intersection of immersive media design, experimental data visualization, and interactive artificial intelligence art. Her work's been shown across the globe in juried exhibitions and recognized through a number of prestigious international awards, including ACM SIGGRAPH, Red Dot Design Award, Ars Electronica, and the A Design Award. Weidi is currently a collaborative artist with the Harvard University's FAS CAM Lab and is in residence with the Society of Arts and Technology in Montreal. She holds a PhD in Media Art and Technology from the University of California, Santa Barbara. So you're going to be showing your slide set, right, Weidi? So we need to promote
Yes.
you to do that.
Okay.
Okay. So you should be able to share screen. Can you share screen?
Yes. Let me try with my keynote.
And if you're doing audio, make sure that you have your audio selected.
Okay. Am I sharing? Can everybody see my screen?
We're seeing it. Yes.
Okay, great.
You might like to put it in presenter mode.
Yeah.
Perfect.
Okay. Should I start?
Yes, please.
Okay. Thank you so much for introduction, and thanks so much for inviting me. And this is my second time joining this amazing event. Super excited to share with you guys one of my recent project, interactive AI art that talks about the interbody self as a montage. And currently I'm in China, so I'm using VPN. I don't know, like the connection. If the connection is not stable, please apologies for that. Okay. So I am assistant professor of immersive experience design at Arizona State University, and my research at the intersection of interactive AI art, immersive art, experimental data viz, and I collaborate with AI to do a lot of AI and also participants to make the immersive experience ever changing and evolving. And today I want to share interactive AI art discovery collection. And this is in collaboration with Roger Luo. And we have been showing this place in different international museums, venues. And this project was born out of witnessing my grandmother's memory regression due to dementia. And it's kind of like very upset story. So I witnessed her cherished story, cherished memory dissolved into fragmented language. And it's in 2022, I come up this idea to probably think about a way artistic approach to transform fragmented language to visual stories in real time. And so I started researching in like dementia, and I realized that over 55 million people face this painful reality of memory regression. And it's also in 2022, the release of text-to-image model, AI model that offers a new perspective, really cherish the power of language. And I think it can possibly weave what time and disease have stolen. So here comes to my interactive AI art installation that visualize fading memories in real time. So I want to share a video first.
Giving birth to my son was the best thing I ever did.
I took a cross-country road trip with my friend without any final destination.
초등학교 때 나무 아래에서 친구들과 도시락을 먹었다.
Cuando mi mamá y hermanas viajes en jugar fútbol en el parque.
所有东西都在摇晃，所有东西都在掉下来，地板裂开。
I remember going to Disney World for a Christmas party and seeing fake snow fall from the sky.
Okay, I'll stop here. I hope the video is not too choppy. And I also have this video on my website. So if you're interested, you can visit my website. So basically, I started with design questions related to remembrance, related to human memories, and also imagination. And I also published a paper related to the design methodology and conceptual development, technical methods about this project. And one of my co-author on my research paper, she's from medical humanity fields, and we have a really long conversation about this topic. And she helped me to develop the medical humanity research background to this project. And we realized that in the medical humanity fields, researchers started to investigate the way instead of like to restore or repair our memories, we see memories as an interbodied self. As a montage, and it's kind of evolving, it's kind of updating, keep changing. And so that's one of the inspiration to kind of investigate how to use AI to reimagine memories instead of to repair or restore memories. And this is our system map. Basically, we invite participants to speak one sentence about their fading memories to the microphone. And then we do speech recognition to transform the voice input into text. And then we do text auto-completion to fill in a lot of details to the narrative to make a complete story. And then we use Stable Diffusion to visualize the story. And specifically, we fine-tuning the Stable Diffusion on Alzheimer patients, their visual memories, captions from their documentaries. So the generated visuals is more nostalgic. It's more about love and loss. It's intentional. And we also use a list of image processing techniques to transform the images into the real-time animation, and got inspiration from 20th century art movements, such as printmaking methods and slit scan photography. So I use optical flow, feedback system, pixel shifting to create this kind of visual representation of fading memories. And it has been shown in conferences such as SIGGRAPH, ACM SIGGRAPH, and also in museum settings. So one of the recent exhibition, we adopted this circular shape, and we drew inspiration from observing people gazing at river water reflection. So they often see like human bodies portrayed in the ripples, and it create really moving and abstract reflection. And I think this is an interesting way to integrate memories. So we look into these reflections, we see ourselves as being reimagined and reflected in real time. So here's some documentation images of the exhibition. And also we, this is a very inclusive art exhibition. So we facilitate the AI systems support more than 98 different languages. So we always encourage the participant to speak the language they feel most close to their heart. So we want to create that kind of intimacy and started from the personal stories and extend to the collective memories. Yeah, we want to build the connections between participants, also between humans and machines. So the most current version is an immersive version. And so we have the 3 projectors to do the projection mappings on the curved screens. And it is currently shown in Budapest. Okay, so we also present our papers in medical humanity conferences. So this project blurring the boundaries between remembering and imagination. And we want to use this project to question and reflect on the imagining collective memories for cultural production and reproduction. And thank you so much for listening. Here's my website, our Instagram and email address.
That's fabulous, Weidi. That's beautiful, haunting. Thank you so much for sharing.
Thank you.
We'll move on to our next panelist, Yoon Chung Han. And she is an interaction designer, multimedia artist, and researcher in biometric data visualization and sonification, new interfaces for musical expression, and mobile user experience design. She's an associate professor for graphic design in the Department of Design at San Jose State University. Her works have been presented at many international exhibitions, conferences, awards, and academic journals such as ACM SIGGRAPH, Japan Media Arts Festival, Media City Seoul, ACM Multimedia, Kai, and Leonardo. She was the Art Gallery Chair for SIGGRAPH 2023 and is the recipient of the Adobe Design Achievement Award and Information is Beautiful Award. She holds a PhD in the Media Arts and Technology from UC Santa Barbara. So you were going to share your screen as well, right?
Yes. Just a second. All right. Do you see my screen?
I see your screen.
Okay, awesome. Well, thank you so much for introduction. And then it's really honored to be here one more time. I think I participated last year, but thank you for having me on this the second time. So today, I heard that this will be about the multiscale human body. So I chose some of the project is about the macro human body part. So today, I'll actually introduce some of the projects I've done. So in the museum settings, so I've been working on various topics related to human body. So first one is a biometrics. I was very intrigued in the human body's, data, like a naturally created data, which is biometric data. So fingerprint, iris, veins, it actually includes many interesting information. So genetics, and of course, for example, our eye color actually comes from, your parents, grandparents, or relatives. But at some point, there's a kind of randomly created like a function incorporated. So there's kind of unexpected things also happening. So I think it was really interesting part. So I want to actually learn more about it by investigating like details of it, in more artistic and experimental setting, and then by using also digital media technology. So this is the first project I've done called the Disty Sonus. So it's an interactive fingerprint sonification art. So this one actually, you can actually scan any finger and then it actually recognize all your line formation. And then basically your fingerprint will turn into like a musical note, and then you can actually create your own sound sonification. So, you can actually create some kind of your own sonic signature from the fingertip. So I think with the fingerprint is somewhat kind of visible, but not very clearly visible. I was very interested about that part. So kind of a little micro part, but it kind of expanded to like sound, different medium using technology. So that was the one I tried it. And then part of the series, the biometric data art series, this is eyes. It's an interactive iris sonification art. So pretty much the same interaction. So you scan your iris, and then actually the eye actually includes many interesting colors. Everyone has you might have a similar brown colored eyes, but the brown itself is very different. So you can actually, the system can actually detect the different color extracted like pixel data from your eye, and then actually turn into the sound. So you can actually also have your own sonic signature. And also at the same time, like 3D visualization on the screen as well. So like it's a multi-dimensional work. And then as the also another part of the biometric data artwork series, with this one is called the Rose and You. So this is a little bit different approach. So this is about the veins. So it actually the system actually recognize your vein lines, as a in the part of your body anywhere, and then try to match to the roads or streets or valleys in the Google Map API, and then try to find where is the most closest, the kind of most similar, the line and then actually streets or valleys or roads in the world. So your vein might actually look like some kind of alleys in the Paris or India or Africa. So it's kind of interesting approach to find the correlation between two very different, data. And then what is this kind of question is about what is the similarity and difference. And then the vein data is actually under our human skin, but the roads or streets are behind, outside of our skin. Because I was kind of curious about data inside and outside. This is not related to human body, but this is about like some kind of hypothetical apocalyptic, near future scenario design I recently created. It's about the microplastic issue. So I actually had a chance to show this work in the many immersive rooms in Korea or other places. So it's all these particle animations are represent the microplastic. So it's actually kind of sceneries you can see in this immersive room, but you can see there's a kind of little weird like plastic objects that are actually kind of decomposing and then turn into like particles. You can be actually part of this scenario when you actually enter this room, and then you can actually experience it, like what will be actually kind of future scenery, when we actually are all covered with the microplastic. So it actually doesn't look like a very dark or apocalyptic. It's in a very opposite way. It's very bright, and then kind of at some point it's very beautiful. So that might be really like maybe reality we might actually face in the near future. And the last project I'd like to share is the It's totally different topic, but I thought it's probably really related to this this event. So I was very interested about the chromosome, human chromosome data. So chromosome is very important part in our human body. So it actually is in our cells, and it actually contains the genetic information, right? And then there's 23 pairs, and then total 46. And then 22 actually determines like very kind of genetic things, and then the last one determines the gender. So and then I had actually had a chance to kind of do some like a PGT test, like the kind of analyze the cells and then kind of extract the DNA, and then it's very interesting to see the result. And then it's actually pretty much like based on my personal information, the personal experience. So, but in the actually, there's also kind of ethical issue as well. So, everyone want to have a perfect baby, and we want to be a perfect, so, and the people actually, to do so, people actually try to really analyze it, understand the chromosome first, and then maybe some actually also because of the very rapid the improvement and development and the medical science, they actually sometimes actually manipulated the genetic information or chromosomes to make a perfect human. So then what would be the meaning of the perfect human, and is it any like an ethical issue or something? So that was actually a question I had in this project. So it's pretty much like I also collaborate the AI. So the images are 3D assets are generated by AI. I collaborate with AI to generate some stories about all these hypothetical 23 babies. So based on the characteristics of each chromosome, each baby has their own story. So yeah, and then it's actually interactive art installation. There's a touch screen panel. You can actually click each individual button, and then you can actually observe the different kind of interestingly designed 3D baby. So each one has a very interesting story. So yeah, here's the video. So for example, one chromosome actually is about like water, because it's actually related to some kind of our veins and then hormone system. So actually I focus on that part and then kind of added some kind of water part, or this is actually control like obesity, like weight control. So yeah, so each chromosome driven, like the baby can actually talk about, kind of raise up questions. What is a perfect human? And then we all actually come from those kind of small cells in the chromosomes, but what will be the thing we can actually, what will be the future human design with the new media technology? So that was this is also exhibited in the currently in Busan Museum of Art. So and then the viewers can actually understand like various possibilities of the human birth, future methods of the genetic modification, ethical dilemmas, and the relationship between future humans and then nature and the environment by observing this work. So the last one is actually, it has a color, and then this is very special one because it's driven by the XY, the gender chromosome. So it actually includes the colors, and then the other designs actually are almost like a black and white color. Okay, thank you so much.
Thanks, Yoon. That's really fabulous. And I can't wait to see more of it, but thank you for presenting. And our next panelist is Joe Heimlich, who's a Senior Director of Research for the Center of Science and Industry in Columbus, Ohio. He's a researcher for its Center for Research and Evaluation, and also a Professor Emeritus at Ohio State University. As an educational psychologist, he's focused his career on how people learn in informal settings like zoos, nature centers, parks, museums, science centers, and other contexts. He has worked in the environmental learning field for over 40 years as both an educator and researcher, and is the author of over 100 academic journal papers and hundreds of other publications. He holds a PhD in ed psychology from Ohio State University with a focus on adult learning. So take it away, Joe. And I guess, do you want me to present or do you want to control
Yes.
the show?
If you'd advance, that'd be great.
Sure. I will share my screen now.
All right. So I'm going to go a very different direction and talk about visitors and the idea of scale in a museum, trying to understand scale in a museum. And so some cautions we might want to keep in mind as we're thinking about experiences and scale. Go ahead and advance. For example, we know people are really bad at reading pie charts. They're bad in general at estimating proportions. People tend also not to be able to deconstruct complex visuals, although they can make meaning of them when they see the layers as they're built, or if someone helps them unravel the different layers. Next slide. Generally, people are weak at estimation in general. So next slide. Estimation skills usually are contextually based as people tend to use them. So what I'd like to do is look at three different examples of estimation and scale and explore what the problems might be for visitors trying to make meaning from our either very large scale or very small scale comparisons and discussions. Next slide. Let's start with, well, we're going to look at weight, distance, and height. And let's start with distance. Next slide, please. The circumference of the Earth is 24,901 miles, or 40,075 kilometers. A US football field is 100 yards long, or 91.44 meters long. It would take 1,370,000,000 football fields to circumvent the Earth. Next slide. So that could lead to a wow response. It's huge. Well, that's great if all we want is a wow response. But if we want people to understand the sheer scale of that, the question is probably more like, huh? And the reason is because just how far is that huge distance? People have a problem knowing how far they've walked. Very few people can estimate walking a mile or walking 2 miles or walking 100 yards. We don't know how far it is. And that doesn't even include different kinds of terrains. 100 yards in a dense forest, or 100 yards up a steep mountain, or 100 yards walking on water are all very different in terms of what it feels like. So if we talk about distance, how do I feel in a distance? And how does that translate to my understanding of the vastness of distances? Let's go ahead and go to the next slide. The weight example. African American elephant is the largest land mammal in the world. They can weigh up to 7 tons. An average adult male in the United States weighs 199.8 pounds, 200 pounds. So the elephant is equal in weight to 70 adult human males. How heavy is 70 adult human males? I can see, and most of us could imagine, a room full of 70 men at that height and weight. But we cannot grasp what the collective weight would feel like. Think about how routinely you lift 200 pounds to compare objects. Basically, with weight, when it's exceeding the weight I can lift, it doesn't matter how much more it weighs than it does, because I can't imagine that moreness. And that's always our challenge is how do we help scale make sense to someone when we're limited by our own senses? Next slide. Height. People are not good at estimating height. One of the most fascinating things in a lot of studies is that in a room, people underestimate the height of people who are as tall as they are and overestimate the height of people who are taller than them. Men especially underestimate the height of other men, and women in the US, women tend to overestimate the height of men. in a room. Now, let's go to a bigger example. When, let's say, witnesses to a crime or witnesses to an incident, and there have been a lot of studies constructed to try to find out how accurate are people in these estimates of somebody's height. What we find is that on average, when you have a lot of people estimating, it's pretty close. But what's really interesting is in all those studies, it's a bimodal distribution. One mode is below the height, the actual height, and one mode is above the actual height, suggesting that most people are wrong. And they're overestimating or underestimating the height of the person. So then let's look at something like skyscrapers or buildings. At distance, we can get a relative height of things and make a visual comparison of taller or not taller. But the reality is when you're on a street in front of a skyscraper and you look up, they're all tall. And we don't have a real difference of height. Trees present yet another challenge, or nature environments present another challenge, one of which is we are terrible at estimating heights of objects in nature. Part of it's our distance, part of it's the shape. Part of it is we don't have anything for comparison with the tree. So we're making a guess, and unless we're using the tools of foresters and forest industry folks about estimation of trees, we're usually really wrong on those. Next slide. So what I'd like us to think about is the language we use with people and the scale concerns. We use terms such as really tiny or really small and really big or very large. Think about these comparisons. Really small town. Few residents, not much happening. We call those very small towns. But then how big is a town in comparison to an atom? Which is also very small. So when we're using those labels, how are we helping people bridge from their experience of very small to what we mean by very small? And the same with really big or really large. Shaquille O'Neal is one of the largest people on the planet, tallest, but he is very small compared to the size of Jupiter. So the language we use and scale is all relative to the comparison object. And having challenges with that is what we will face as we create these exhibits or opportunities for making meaning of scale in museums. Next slide. So a couple of other little cautions. Language we use as experts often does not translate into the vernacular of people. As a human, knowledge I hold means something. And at scale, knowledge a cell holds. I try as a human to then say, where's its brain? Rather than understanding it may be chemistry, it may be electricity. We don't, using the labels that we have in our vernacular can often lead to confusion when we're talking about something technical. Same thing with an environment, with trees and the fungi and other plants that they can communicate with. When we say they communicate or they talk, people automatically put into their minds that they're talking about words and language as we know it, and communication through sensory things as we know them. So helping people make these bridges becomes very important in understanding scale. And last slide. Should be one more slide, Stephen. There. Yeah. So the bottom line on these, does scale matter? Or does the information we're trying to convey about scale matter? Sometimes that extreme example of very small or very large is not helping us when we want the information to be about relationship. So understanding what we're trying to communicate becomes important. Provide a constant comparison of scale. Something that we can hold on to as a human that helps us see how far down or far up we're going with our scale. Use language carefully. Think about where else and how else a descriptive term is used by the potential visitors. Acknowledge that beyond scale, on certain points in scale, both up and down, people cannot make a distinction. When they've reached their limit, it doesn't matter how much further you go, out or in, they won't understand it as more or less. And that's okay. People don't need to have the absolute of a scale difference. They need to have a relative understanding when we talk about scale, when we're illustrating at scale, to make meaning from it. And that's our job as educators in institutions and as communicators of science. Thank you so much.
Thank you so much, Joe. Looks like we have our work cut out for us. Thanks so much for that presentation. I think we'll move on to Katy. And you probably know most about Katy as our final panelist, but in case you don't and you're new to the webinar, she's the Victor Ingve Distinguished Professor of Intelligent Systems Engineering at Indiana University and the founding director of the Cyber Infrastructure for Network Science Center. She's also a Humboldt Fellow and a Fellow of the AAAS and the Association of Computing Machinery. But Börner has written over 200 articles in academic journals and scholarly texts, edited several publications, and is the author of the ambitious Atlas series of books on data visualization and analysis. She earned her PhD in computer science from the University of Kaiserslautern. She's a true transdisciplinary thinker and collaborator and a very creative person. And thanks, Katy, for being a leader of this great adventure. And you can share a screen now.
Yeah, very nice. Thank you for having me here, Stephen. I think we connected in one of the IM2 meetings way back in time, where actually many researchers and science communicators and publishers and funders and others came together. And it was a wonderful, marvelous event. That was the very first time I got to talk with Science Museum and other museum experts. I learned a lot, and that was the gateway, the opportunity to bring some research into these public spaces. And we also have brought research into libraries and into many, many other places, because it matters. It's important that it's not just staying in the ivory towers in the research environments, but it's really, really important that many more get to be excited about science, become scientists maybe even, but also just know about the power of science. And so oftentimes we try to collaborate with artists, and that's not always easy. In some cases we get a blunt no, we don't want to collaborate with scientists, but in other cases we find collaborators that make new experiences out of the data and the tools that we have created. And so here you will get to see an exhibit that was inspired by HubMap, the Human Biomolecular Atlas program that has generated a lot of spatially explicit data sets. And they're just beautiful, but they're all digital. And we wanted to make them more touchable, hands-on, so that people get touched by them a little bit more. And so here in the next slides, you will see some of the pieces, and they are also on display in Luddy Hall, our work environment. And here on the left-hand side, you see a sculpture of science that looks a little bit like a mushroom, but you can actually trace the evolution of science using these little marble balls. And then you see some other elements, which I will introduce to you. And so here the first piece is by Carrie Longley, which also did the sculpture of science, but what you see here on the right is what I call an alien baby, but she calls it Capillus linus, hairline. And this piece was made from clay, wire, pig intestine, and wax. And it shows a very playful shifting between our traditional ideas of what scientific specimen look like and what an art object looks like. And actually over time, it kind of deteriorates, and that's part of it, because life does deteriorate over time. We are born, we grow, and then at the end we go. So I think it's really capturing a little bit of the mystery of our natural world. And Carrie is a great studio artist and educator. and professor of fine arts at Indiana University East. She has a great website. So if you want to see more of these Aryan babies and others of her work, please go there. Another piece that we were able to do with an expert in biology, assistant professor, newly hired assistant professor in biology, Dr. Luke Nikolov, is tabula floris. So here, instead of looking at single cell data of the human healthy adult body, you see flowers, and you actually will also see single cell data of these flowers, not shown here. But it's super interesting to realize that it is much, much easier to get tissue or materials from flowers than it is to get them from healthy humans, because healthy humans typically don't like to give up tissue, except it's maybe adipose tissue or placentas or something like that. And so still the same analysis techniques, the same single cell RNA techniques and analysis and UMAP plots can all be applied to other specimens as well. All right, this is work by Beata Edita Mirswa. I met her at the WISPY conference, which is an amazing get-together of artists and scientists and engineers and others. And she and her partner, they are actually bringing a lot of art to that conference, including also T-shirts and dresses and scarves and socks and everything else. But here you get to see a protein-protein interaction network on the left-hand side that served as inspiration. And then on the right-hand side, you see an artistic rendering by Beata as the realization. And it's always the inspiration here on the left and then the realization on the right. And so you see that some of the artists have been trained in biomedical research, and others come to all of this as an artist, and I think both is super valuable. And I was just astounded to see the works that have been presented before me. Here you see beauty is everywhere stitching of beads by a jeweler, Angela Caldwell, actually here in Bloomington. And her work was inspired by a codex image of the isthmus, a short, muscular, rounded section of the fallopian tube. And she even showed the fallopian tube on the right. It's not shown in the inspiration piece. But the inspiration really comes from a much larger site, which is the image of the week website for HubMap. And you can click on the link, and maybe Meryl can send it to the chat also. And these images are really beautiful. They show human healthy tissue that we all have, at the single cell scale. And the big question is, how can we use machine learning algorithms? How can we use our human intellect to puzzle together from all of that the healthy human being as it lives and breathes and evolves over time? And so again, it's neat to see it rendered in a very different material. And we have a lot of people stopping by the little exhibit, which is really just the beginning of a much larger exhibit we would like to do with all of you. And it really inspires people to say, oh, I can do this. I can go either take this immunohistochemistry image, or I can pick another one from this amazing set of experimental data. And I can make my own drawing, my own stitch work, my own bead work from it. A final piece I would like to show is Cece and Squiggy. They are coming from the scrolly story environment that we have going with HubMap. They are super friendly tour guides introducing readers to a number of core concepts that are associated with the Human Reference Atlas. We realized soon that it is not easy to understand what an atlas is, how you map new data onto it, why in the world do you need to register tissue into 3D organs. And so there's a team of three, Shovik, Melanie, and Todd, which are developing stories and implementing them in 2D and in 3D that help everybody understand terminology, processes, and also the ambition behind the atlasing effort. And so here you see a ciliated columnar epithelial cell, also called CC, that being here, and a simple squamous epithelial cell, squiggy, right here, blue. And we also have a 3D printout of both. And so you see them in the 3D environment introducing different user interfaces, different concepts. But then, of course, now you also have them materialized in our world. So I think it's super fun to do this. And if you go to the QR code here, you will get to the stories, which as you go down the website, you get to experience their stories. And they have very different personalities, and they are very uncomfortable sometimes, but they help each other all the way. So here you have some of the screenshots. So initially it was all 2D, but interactive. So you can, as you zoom down, you zoom in. However, now they actually exist in 3D together with their friend macrophage, MAC for short. And so I think it's a fun and easier way to introduce all of this than having long scrolling standard operating procedures. So this is all I wanted to show. There's also the vasculature common coordinate framework video, but we already had that shown in the Illustrator panel, so I'm not gonna feature this here again. So thank you, Stephen, for hosting all of us, and I will stop this here.
Great, thank you, Katy. So I guess this evokes a few questions that I have for the panelists, and then we'll take some Q&As. For the audience, please put your questions into the Q&A panel, and we'll read them off to the recording and have the panelists address them. But I guess the first question I have, particularly after seeing Joe's presentation, thinking about scale, it's this idea that you have these cells that actually form different things, and in particular organs, I'm thinking, but it could be anything, any set of organs in the body. But how do we do any of you have a sort of a perspective on how we might take that transition from cells to organs, and how would you communicate that? I know it's a big question, but just trying to think of how to take all the incredible stuff we've just seen and think about, okay, is there some conceptual idea that might actually help bring that to life? And anybody can address that.
And just for reference, a kidney is 10 centimeter, and Tarek is already on here, he can correct me. And a cell is typically 10 micron. So that's the scale difference.
It's a lot of cells.
So if you would put a lot of 10 micron spheric cells in a volume, then within 1 cubic millimeter, if there are no empty volumes, you would have 1 million cells. So how do you represent that best? And the entire body, as we know, has 37 trillion human and 40 trillion microbial cells. So it's really a challenge, a visualization challenge. That's why I find it so fascinating.
And I think, as Joe mentions, it's okay. But
just what I think is exciting there is just the sheer hugeness of the numbers. I don't know what they mean. I don't need to. I just need to understand it's a lot. And I think sometimes we get caught up in the preciseness of the numbers rather than just letting them reside as, yeah, there's a lot there, or there's not much there. So I think it's the relative. We can't see the scope. We cannot envision that number. It's too big. But I can know that it's bigger than I can even imagine. And maybe that's all I need to know. And this is where I think art,
right? So we have been trying to at least explode the anatomical structures that exist inside of a kidney, because there are many and they're interestingly shaped. So you can have an exploded diagram of that. So this way you get to at least see what they are and where they are.
Yeah. And in that case, the constant comparison of the real kidney size versus that blow up piece wouldn't matter. That would help me understand the scale at that scale. But I also wanted to say it's this challenge that I think makes art such a valuable contributor to science, because it gives us freedom to be able to talk about things from a visual representation that's different.
So one thing just to add to that. So I think it was Yoon in your presentation, you talked about the plastics. So this idea that all of these plastic particles are everywhere. They're in the air, they're in the water, they're in our cells, they're in our bloodstream. I thought that was a very interesting sort of, I guess, lack of a better term, a metaphor for the smallness of things and where they go because they're small. And I know that invokes things like nanotechnology and all that, but just that sort of evoked that idea of you have these things, these very small things, and they sort of know what to do and where to go. And I guess a follow-on to that would be, what kind of metaphors might actually be useful. And I know, we've kind of dismissed a little bit the idea of using metaphors, but there may be ways that we can think of, like the metaphor of memory that Weidi showed, that distorts the way that we see the world because of loss. Thinking about other ways that we might use that kind of metaphorical, artistic way of looking at it. I don't know if Yoon or Weidi have any particular comments on that.
Yeah, so my work, it was about like a marine microplastic pollution issue. So it was actually a multi-channel animation, and then it's actually, you can see a lot of actually small particles actually flying around and then kind of flowing to various directions, and maybe they kind of pile up on the ground. So what I intended was actually, I just want to look like a kind of beautiful, like a little bit kind of paradoxic kind of expression, in my opinion. So it doesn't look like very pollution, whatever, dirty or dangerous stuff, but more look like a kind of flowers or I don't know, some kind of flying kind of objects. So with a lot of variety of colors, because actually plastic really has a lot of variety of colors, very, very pretty sometimes, So I think that metaphor was the one I tried it. So plastic is like now really part of our life. It's actually everywhere, even not only just the surface of the ocean, but it's there's a study that actually is really in the deep sea now. So you can actually discover plastic in the very, very deep ocean. So I think probably in the near future will be pretty much more like everywhere. So it's also in our body too. Microplastics are in our body. So yeah, I just want to express that this is really just a part of our life now.
Any comment, Weidi?
Yeah, so my practices often started with collecting data from the real world and then transform it into visual representations or sonic representations, and then as cultural artifacts that can be communicated and interacted with participants. So interactivity is one of the key components for most of my piece, and I think interactivity and immersive media can definitely offer a great opportunity to explore scale. And yeah, so for example, there's some artwork, maybe research projects from University of Southern California and School of Cinematic Arts. They created research projects called Wood in a Cell, and people can actually travel in the cell and interact with it. And there's a bunch of like immersive UI. You can kind of adjust and zoom in and out and for different elements, components in the space. And I think that probably could be an interesting way to interact with the cell. And also for my piece, I kind of implement a more artistic approach. So sometimes I really embrace the mistakes or imperfection of from the scientific different from the scientific approach. So I think sometimes the imperfection can provide opportunity for creativity and also kind of surreal approach to create emotions. So I really enjoyed that. So it's kind of like different twist of difference between the artistic approach and scientific approach, but sometimes I really enjoy it. Yeah.
Yeah, well, there's a question actually in the Q&A, which actually it relates to that. So Vivian asks, good communication is a two-way street. For the artist on the panel, how do you experience working with life scientists? Do you, for example, get the impression they also learn from you and or enjoy the interaction? And that's from Vivian Marks, journalist, Nature Portfolio. So do either of you want to take that on? If you want to read it, it's in the Q&A.
Okay.
Working with scientists, do you have a particular perspective on your experience working with scientists?
Yeah, I'll just quickly mention, I actually had a lot of collaboration with engineers and scientists before, and then what we actually see is very different, you know. So there are a lot of actually kind of sometimes troubles or fights, you know, but which I consider as like a learning process and then creative process. But I think the technology and art are, they actually go together. They are developed together. And because the technology is actually really like represent what we actually are seeing in current age. So it actually really includes our cultural aspect and then societal aspect as well. So how we're gonna actually apply into our life, I think that's the role of an artist, in my opinion. So artists can actually put more like add more narratives, you know, stories, how we can actually use the technology to have some kind of meaningful experience for the viewers about a certain topic. So I think the collaboration is very very important, in my opinion.
Yeah, and we're collaborating with one right now. Well, actually three, if you count the various kinds of science. So yeah, I guess one thing that brings up for me is we have there are a lot of aspects of things like how cells become organs, how cells become limbs, what the morphology of living things is versus what cells know, how they communicate, you know, morphogenesis. There are a lot of very complex ideas, and thinking about how do we get across some of the ideas that actually say how this works. You know, how does cells, you know, start off with basically one cell? How does that turn into what we see as a human being, or a cat, or a moose, or a bull elephant? You know, there's a lot going on that is very hard for people to grasp, and it's just very interesting that that could take a whole night just talking about that. But I don't know if Katy, do you have a sort of a take on that, or anyone in the panel?
I think we absolutely must collaborate across boundaries, across ages, across funding organizations also, right? We had publishers earlier, we had funders earlier, and I think there is a growing consensus that we can't do it alone. It's a large village effort that needs to get done here, and we all will benefit, and it's actually more fun this way. So again, Weidi, Yoon, I look forward to talk more. I would love to see you embrace that cool data that has been generated in HubMap and in many, many other projects, and it's super visually stunning. And again, I think two of the experts that are actually generating this data are now joining, so Tarek and Daria, and I'm looking forward to have them explain a little bit of how you actually get to this beauty, right? How do you zoom into the single cell data and help us all understand how it is structured, what the function might be, what happens when disease comes in. And it takes this kind of mixture of different minds and different views of the world to communicate that to us and to others, and to ultimately also bring in AI in meaningful ways. So Stephen, thanks so much for leading this panel. I truly enjoyed it. And total pleasure. It's always good to have your wisdom on the board and on the panel here. It's really, really nice to see you all here.
Yeah, thank you, Katy, and thanks Joe and Yoon and Weidi for taking time tonight to look at this. This has been fantastic, and we're handing off to the next group. So thank you, and have a good night.
Yeah, and nice, Stephen, I like your ambition. You know, how would you make a museum exhibit out of all of this? And I think it will take stunning visuals and things that really resonate with people. And artists are just good at getting us to resonate to art pieces, but also with each other, ideally together with the art piece. Thank you for that.
Okay, thank you.